inoculum preparation is very crucial step in the fermentation industry. So friends, let's learn this topic. This discussion, the development of inocula for yeast processes, is a part of NICT project, entitled Creation of E-Contents on Fermentation Technology. Sponsored by the MHRD, Government of India. Industrial uses of yeasts are 1. The brewing of beer. 2. The production of baker's yeast biomass and 3. For the production of recombinant products from the yeast. Brewing. Yeast used to inoculate a fresh batch of wort from previous fermentation or from propagator. It is common practice in the British brewing industry to use the yeast from the previous fermentation. The brewing terms used to describe this process and crop, referring to the harvested yeast from the previous fermentation, and pitch, meaning to inoculate. One of the major factors contributing to the continuation of this practice is the wort-based excise laws in the United Kingdom, where duty charged on the sugar consumed, rather than the alcohol produced. Thus, dedicated yeast propagation systems are expensive to operate because duty charged on the sugar consumed by the yeast during growth. The problems with this technique are chances of contamination and degeneration of strains. The most common problem with a degenerated cell is the change in the degree of flocculence and weakening of abilities of the yeast. In breweries employing top fermentations in open fermenters, these dangers minimized by collecting yeast to be used for future pitching from middle skimmings. As the head of yeast develops, the surface layer which contained the most flocculent and highly contaminated yeasts removed and discarded, and the underlying cells, that is, the middle skimmings, harvested and used for subsequent pitching. Therefore, the middle skimmings contain cells which have the desired flocculence and which have been protected from contamination by the surface layer of the yeast head. The pitching yeast may be treated to reduce the level of contaminating bacteria and remove protein and dead yeast cells by such treatments as reducing the pH of the slurry to 2.23, washing with water, washing with ammonium persulfate, and treatment with antibiotics such as polymyxin, penicillin and neomycin. However, traditional open vessels are becoming rare, and the bulk of beer brewed using cylindro-conical fermenters. In these systems, the yeast flocculates and collects in the cone at the bottom of the fermenter, where it is subject to the stresses of nutrient starvation, high ethanol concentration, low water activity, high carbon dioxide concentration and high pressure, which decreases the viability and physiological state of the yeast crop. Would not be ideal for an inoculum. The situation is further complicated by the fact, which the harvested yeast is stored rapidly to about 1 degree before it is used as inoculum, suspending in beer and storing in the absence of oxygen. One of the key physiological features of yeast inoculum is the level of sterol in the cells. Sterols are required for synthesis of membrane, but they are only produced in the presence of oxygen. Thus, we have the irregularity of oxygen being required for sterol synthesis, yet anaerobic conditions are required for ethanol production. This irregularity is resolved traditionally by aerating the wort before inoculation. The difficulties outlined above and the likelihood of strain degeneration and contamination mean that rarely used for more than 5 to 10 consecutive fermentations that necessitates the periodical production of a pure inoculum. Pure inoculum prepared by a yeast propagation scheme, utilizing the 10% of inoculum volume at each stage in the program and employing conditions similar to those used during brewing. Continuous aeration used during the propagation stage, which seems to have little effect on the beer produced in the subsequent fermentation. Yeast inoculum produced in this way would also be sterol-rich, obviating the need for aerated wort. The simplest type of propagator is a single-stage system resembling an unstirred aerated fermenter, which inoculated with a shake flask culture developed from a single colony. Two-stage systems propagator operated semi-continuously. It consisted of two linked vessels, 1.5 and 150 cubic decimeters respectively. The smaller vessel filled with wort, sterilized, cooled, aerated, and inoculated with a flask-grown culture. After growth for 3 to 4 days, the culture forced by air pressure into the second vessel, which, filled with sterilized, cooled wort and aerated. An aliquot of 1.5 cubic decimeters, forced back into the first vessel after mixing. In a further 3 to 4 days, the larger vessel contained sufficient biomass to pitch 1,000 cubic decimeter fermenter, and the first vessel contained sufficient inoculum for another second stage. However, although this procedure should produce a pure inoculum, there is a danger of strain degeneration occurring in such a semi-continuous system. Brewing. Yeast used to inoculate a fresh batch of wort from previous fermentation or from propagator. It is common practice in the British brewing industry to use the yeast from the previous fermentation. The brewing terms used to describe this process and crop, referring to the harvested yeast from the previous fermentation, and pitch, meaning to inoculate. One of the major factors contributing to the continuation of this practice is the wort-based excise laws in the United Kingdom, where duty charged on the sugar consumed, rather than the yeast pumped from one stage to the next. The seed cultures may be centrifuged and washed before transfer, which reduces the level of contamination. The yields obtained in the first 5 stages are relatively lower because they are not fed batch systems, whereas the last 3 stages are fed batch.